Hi, my name is Zoe, and today we will be talking about how SARS-CoV-2 translates its genome into proteins.
To convert its genome into functional proteins, coronaviruses use two main mechanisms. First, they produce individual proteins in one long chain that are processed and cut after being translated. The second method involves the production of messenger RNA strands that are shorter than the full length of the viral genome. Let's consider the first option, the production of large polyproteins. The single-stranded positive sense RNA strand from the 5' end contains the code for 16 different non-structural proteins, commonly referred to as NSPs. They are involved with a wide range of functions, including blocking the host cell's initial immune response and contributing to and forming the viral replication and transcription machinery. These 16 proteins are encoded for within two open reading frames, labeled ORF1A and ORF1B. An open reading frame refers to the triplets of nucleotide bases that the ribosome reads and creates the proper chain of amino acids from. There is a stop codon in between the two open reading frames that tells the ribosome to let go of the mRNA strand and release the polypeptide chain. If there were no mechanisms to get around this stop signal, only the proteins in ORF1A would be produced. However, for the three nucleotides in a row that mean stop to be acknowledged as the stop signal, they need to be in frame, that is, read as a triplet. The coronavirus RNA has a frame-shifting mechanism based on its structure and sequence in between ORF1A and 1B that allows for the production of proteins in ORF1A and 1B in one long strand. This mechanism occurs by chance, keeping the ratio of proteins in each ORF in the correct proportion. To free all the NSPs from their long chain, NSP3 and NSP5 contain viral proteases, which are enzymes that act as molecular scissors, to individually free all the NSPs that were transcribed in one long chain. This phenomenon of producing multiple proteins in one long chain does not normally occur in human cells. However, there is still 1/3 of the viral genome left, containing the structural and accessory proteins that must be translated into proteins as well. This is where the transcription of what is called subgenomic mRNAs come into play. Recall that RNA and DNA are transcribed in the 5' to 3' direction. Thus, when the coronavirus positive sense RNA genome is transcribed to negative sense RNA, the transcription complex begins at the 3' end, where all the structural and accessory proteins are encoded for. As the complex moves along the positive sense RNA genome, it comes across transcription regulatory sequences referred to as TRSs. At these TRSs, occurring at the 5' end of every individual protein's gene, besides those in ORF1A or 1B already described, the transcription complex is able to pause. Then, it has two options. It can keep transcribing its negative sense RNA strand, or it can jump to the leader TRS sequence all the way at the 5' end of the positive sense RNA genome. By jumping to this TRSL sequence, the complex has made a negative sense subgenomic RNA strand. This RNA strand will be used as a template for a positive sense mRNA strand that will be read by ribosomes and translated into the correct protein. Now, because of the transcription complex being able to jump to the TRSL sequence, the first gene on the subgenomic mRNA can be any one of the structural or accessory genes, thus allowing for all the proteins the coronavirus encodes for being produced. This concludes the explanation of how the coronavirus's single-stranded positive sense RNA genome is translated into multiple proteins. If you enjoyed this video, we encourage you to check out the other Biomaker TV products. Thanks for watching!